So we've gone through the law of dominance or principle of dominance and the principle of segregation. Mendel's third law requires a look at, instead of a monohybrid cross, a dihybrid cross to get at the principle or law of independent assortment. So what does that mean? Well, we know on our chromosomes that we have different specific genes, maybe for hair color, eye color, all the different traits that you can imagine are carried in specific spaces on specific chromosomes. And so what it tells us is that when our gene pairs separate, they are also the genes from our, say, eye color gene is going to separate differently from the hair color gene. So during meiosis, members are a pair of genes on homologous chromosomes. They're getting distributed into gametes independently of other gene pairs. So eye color versus hair color getting distributed independently of one another. So how did they determine this? How did Mendel determine this principle? As I mentioned, he did what's called a dihybrid cross. So a monohybrid just means one trait at a time. You're looking at a hybrid for one specific trait. Here you have a double hybrid, so a hybrid for two traits in the same organism. And trying to see what the probabilities would be if these organisms, these dihybrids, were crossed, that you would have these different phenotypic outcomes. So he started with pure breeding dominant parents for one, a pure breeding dominant parent for one set of traits, two different traits, and then a recessive pure breeding for two different traits. So by crossing those, you end up getting a dihybrid. So if you look, this parent can only donate capitals. This one can only donate small letters, meaning that the offspring, regardless of which P or which T gets transferred, they are going to end up dihybrid. And that gave him the famous 9:3:1 phenotypic ratios that you've probably seen on the large Punnett square. So if we look at the possible gametes from our dihybrids, these are the four possible pairings of T's and P's. And it's exactly the same because you're crossing an identical dihybrid other parent. When you fill in the Punnett square, what you find is purple is the dominant trait, and it happens in a 3-to-1 proportion to the white trait. And then height, tall or short. So these are the short ones, 1 short, 2, 3, 4 short out of 12. So again, 3-to-1 on the height gene. So does independent assortment always hold true? It only works for genes that are on different chromosomes. So it can occur if genes are on the same chromosome, if those genes are far enough apart on the chromosome. Because if you remember, crossing over allows some switching of genetic information from one homologous chromosome to the other. And if crossing over happens with genes that are close to each other, they might often cross or be crossed or paired together. And but if they're far away, they would not be. So if they're very close together on the same chromosome, they are considered linked genes because during crossing over, they might not totally assort themselves independently. It just so happened that the traits Mendel was studying in his dihybrid crosses did happen to fall on either separate chromosomes or far enough apart that they did sort themselves independently. So all genes on one chromosome are considered a linkage group. And again, the farther apart two genes are on the chromosome, the more often they cross, the more often crossing over occurs, which could separate them. So they'll be more likely to assort independently of one another. And so using probability with offspring, crossing parents and looking at the outcomes that certain alleles show up together or separately, that probability concept is what Thomas Hunt Morgan used to develop chromosome maps and studying fruit flies. And we're going to do a simulated fruit fly lab to try and see if we can make sense of that idea. So if you look at these diagrams, gene 1 and gene 2 are very close together. They're linked. And very likely they will not separate themselves independently because they're so close that if crossing over happens, they would often go together. These are on separate chromosomes, so they're not linked. And then these are far enough apart that they might be in the same linkage group, but they may not be linked according to an independent assortment. Okay, so do Mendel's other principles always hold true for homologous genes? So principle of dominance, principle of segregation. Well, dominant and recessiveness is one that's tricky because not all traits follow those rules tightly. So expression patterns of genes for some traits are not as straightforward. We will learn about, or you will read about something called codominance in the chapter, where two alleles that are expressed fully gets expressed in the heterozygote, so that the phenotype can sometimes be a double expression of protein or two different phenotypes, or a sort of a blended phenotype expressed. Then there's multiple allele systems, which we'll explain in a little bit with some examples. But that means you have genes that have 3 or more alleles in a population. So it's not just purple versus white. There might be a whole bunch of different ones. And sometimes you have both of those together, codominance and multiple alleles. So in the case of blood type, A blood and B blood are both codominant. It's immunoglobulin A versus immunoglobulin B. There's 2 versions of it, and if you have AB blood, that means you have both alleles and both proteins get made. You can have a recessive allele for AB blood type, and so your immunoglobulin might be a recessive, and that would lead to O blood type. And so the multiple alleles are the recessive O variation, the B variation, and the A. So you have a multiple allele system here, 3 different alleles for immunoglobulin, and then you have 2 that are codominant, A and B. If 2 alleles are not fully dominant over one another, that's something known as incomplete dominance. Your book gives some examples of that. I think they use snapdragons as an example. But the heterozygote's phenotype is somewhere between the 2 homozygotes. And so you can see, snapdragons was the example. Big R is red. Big R, little r is pink, the heterozygote. And little r is white. And there's the 3 different phenotypes that can be produced from incomplete dominance. And so the heterozygote is sort of a blend, again, of these 2 phenotypes. This is just a Punnett square showing the phenotypic outcomes of the different genotype possibilities of the offspring in that Punnett square. And straight out of your text. So what's it called when more than one gene influences a trait? So genes code for proteins. That means that multiple proteins are applied to forming the trait that you're looking at in this case. And they call that epistasis, or they would be called epistatic traits. So you get 2 or more gene products that actually play into that expressed trait that we see. So sometimes one gene can suppress the effects of another. And coat color in dogs is the example from your text. The yellow lab versus the chocolate lab versus the black lab. And I think there's 3 different alleles at play. But one that I think is probably more interesting for us as humans is eye color. So we always talk about brown eyes versus blue eyes, but we also look at quite a variety of different types of blue eyes or types of brown eyes. You have green eyes and hazel, and these different, maybe more amber colored. And what's so interesting about that is that, well, why is it not just all or none? Why is it not just brown or blue? It's because your eye color is actually an epistatic trait. We simplify it oftentimes with Punnett squares, but eye color is determined by actually 16 different genes that are adding to proteins that play into your eye color. And so you have examples of incomplete dominance in there and epistasis for determining eye color. And this was taken out of a journal article I read in the Journal of Human Genetics about eye color because people were asking about that, so I decided to dig in. So epistasis, a couple different examples, one from the book and then this one from notes on eye color. This slide's just worth it for the picture alone because these guys are all so cute. But this is a Punnett square trying to show you this epistatic interaction to get the different coat colors and what genotypes you'd have to combine to get yellow lab, chocolate lab, or black lab. Another concept that was shown up in the text was pleiotropy. What the heck is pleiotropy? Well, that's where one gene influences many traits. One gene, one protein produced by the body influences many traits in that developing body. So Marfan syndrome is an example of pleiotropy. And so this gene expressed protein results in a variety of different phenotype expressions. All of these things can be affected by that gene. So that's a pleiotropic gene, influences multiple traits. Another pleiotropic example in humans that you get screened for at birth and you can control with a specific diet to avoid symptoms. It's called phenylketonuria, PKU. It's a disorder caused by a deficiency of an enzyme, phenylalanine hydroxylase, which you need to convert the essential amino acid phenylalanine into tyrosine. And I knew a kid in high school who had this, and so he had to just avoid this in his diet, and he was okay. But if you don't, you can end up with a toxic overload of amino acids you can't digest.